Welcome everybody, and we are very happy to show you what we have been working on this last week. The main topic is that many people want to express enzyme. Enzyme in this time of COVID pandemic are extremely important because they are the heart of diagnostic tests and research. So unfortunately, production of enzyme is a long and cumbersome process. So it goes from design, cloning, expression of the enzyme. Most probably you want to purify them, and you want probably also to check if they work correctly, and then possibly storage and sending them around. So we worked these last few weeks to make this process accessible to as many people as possible and make this process understandable and easy to follow and easy to implement. And this is an introduction to what is the research in diagnostic DNA collection. We start with the design, and this is where this DNA collection comes into place. It has been a collaboration of, as Jenny said, different partners, the Open Bioeconomy Lab, Ferdinand Federici Research Group, the Free Genes, and the Vancouver Open Science Network with the supervision of Ginkgo Bioworks. So there were a lot of heads coming together to make this happening, this DNA collection happening. So what is this DNA collection? What will enable you to do? So the DNA parts present in the collection, right now in the collection that will be soon available, is currently being synthesized by Twist, contain 16 ready-to-express cassettes. So are cassettes that we draw from patents and literature that we know they are working. And they're cassettes that are going to express enzymes that usually they are used widely in diagnostics tests, meaning reverse transcriptases, BST for LAMP, DNA polymerases, RPA enzymes, DNA inhibitors, and these are just few of the enzymes available. All these enzymes currently can be expressed by adding a PTG, and the enzyme can be purified by histag. We also added some cassettes for the expression of RNA controls for SARS-CoV-2, 5 positive from different positions in the SARS-CoV-2 genome and 2 negatives from MERS and SARS-1. But of course, you might ask yourself, what if I don't have IPTG? What if I cannot or don't have the columns to actually do histag purification? So we came up with a collection of modules of DNA. So beside giving you the possibility to use these ready-to-express cassettes, and I cannot stress enough, these cassettes are open source and are off patent. So all these enzymes can be used for research and in a company anywhere you want. Anyway, if you don't want to use histag IPTG, in the collection are available modules to construct your own expression cassettes tailored to the resources that are in your lab or wherever you are. So which are these resources? So we have added different enzymes connected to diagnostics. There are up to 20 enzymes in the diagnostics collection that can be used for PCR, LAMP, RT-PCR, RPA, on RPA. And also there are some enzymes that can be used for the detection as well. So horseradish peroxidase, for example, or some reporter genes. And of course, once you choose your enzyme, you need DNA parts to actually express these enzymes. So the most fundamental modules, of course, are promoters and terminators. So we added 6 different promoters, among which we have, of course, the IPTG inducible promoter, but also a temperature inducible promoter. So you can express your proteins just increasing the temperature of your culture. Promoter that is light responsive, so you can change the light and induce a protein, or promoter that, for example, are constitutive. We also added 3 different terminators that have been identified, which are the best terminators that are 3 of those. And of course, one of the things that you might want to do is actually purify your enzyme of choice out of a bunch of other enzymes in the cells. So we also added modules that allows you to fuse your enzyme of choice with a little tag that enables you to purify that. And this is not just histag, but we're talking about silica tag, cellulose tags, for example. So you might be able to use paper pulp to actually purify them. Other modules that we added to the collection are reporters. Specifically in this collection, we added 3 different reporters, but in other collections that will be available soon, we also have 20 up to 28 different reporters. And why is that? Because you might want to add another protein that allows you to see if your protein is expressed and where is expressed, and also maybe follow the purification procedure with the tag through the color or the reporter genes. And of course, at the end of the day, what you want is actually your enzyme. So we also added 5 different cleavage domain that you can add between your reporter, for example, and your enzyme of choice, or directly between the tag and your CDS. And these are cleavage sites that either needs another protein to be cleaved, or are peptide that self-cleave themselves, depending on the condition you put your protein into. And of course, all these modules are interchangeable. So the idea is that if you want either the tag, the CDS, the cleavage, or the reporter in any combination, you will be able to do that. We are just providing the basic playground to actually have these constructs and produce your enzyme of choice. Okay, that's great. We have all these modules. Are we going to stick them together? Are we going to ligate together? There are 2 different ways to do that. And here we are already in the second part of the process, right? We design our construct with all our modules. And once we decide which is the construct we want to make, we have to clone it. And there are 2 ways to clone it. So each of these modules in the collection have already within the module sticky ends that allows you to fuse one part with the other. And what you need to do is isolate the plasmid from the collection, the plasmid that you want, that includes one or the other modules that you picked, and then use BSA1 and ligase in a modular cloning assembly to actually fuse the parts that you have chosen. All these, there are different resources that again are linked to the presentation and you'll be able to access later. We are working towards giving you these tools as well. So in order that whenever you will get the collection, you will have also the enzymes to do this job. You won't need to buy externally these enzymes. The second way to clone these modules is through Plano assembly, which is a low-cost version of Gibson assembly. What you need in this case to fuse different modules is having PCR. You have to have primers of choice, PCR reagents, and then we will provide hopefully also the Plano enzyme to actually do this job. Again, there are a lot of links that you can access to after the presentation. And that's it. I think right now I will give the microphone basically virtually to John, who's also completed parts of this work and will walk you through it the second part of the presentation.
As Chiara said, when or while you are doing your assembly, you will often run it on a gel. And at present, one of the cost restrictions of running consistent agarose gels is the DNA ladder. And therefore, as part of the collection, we will include an open DNA ladder, which, as you will see, runs from 100 up to 9,600 when digested with 2 enzymes, but primarily between 1 and 105,000 when digested with BSA1, which will be part of the collection. And assuming all that goes correctly and you have your construct, you're then going to have to express it. And I've spent many years expressing proteins and know that it can be difficult. Therefore, we've made a large effort to actually try to summarize some of this information into an easy expression guide. This is both work that we've done, but also work that we hope that other people will contribute to. And I know that some in the Reclone forums and chats have actually been adding to it, which is excellent. And basically the overview to this is, if you're wanting to express proteins for particular techniques, we've split it down into particular techniques. Also, you can go down into individual levels, so you can go down to individual enzymes, and within those individual enzymes there are, so for example, for MMLV, there is a quick expression sheet. Now, if you're experienced in protein expression, you can probably simply read that sheet, go, oh, okay, I need to put it in this vector, express it for this long at this temperature with a tag at this end, and away you go. If you would like slightly more information about that, or where that information is derived from, you can look at the spreadsheet, which will detail a non-exhaustive list of sources for the different conditions that you can either create your constructs or express and subsequently purify them in. These are derived from a collection of lab books, both locally and around the world, patents and papers and those kind of things. And then there are selected references. Once you've actually got your protein and you're happy with it, you need to be able to be confident that it actually does what it does. And I've kind of used this kind of report from the Africa CDC and African Union, which investigated the quality systems in clinical laboratories. And certainly if you are testing for COVID-19, or indeed any testing laboratory, it needs to be consistent and you need to be sure of the results. And in this particular case, you can see that the most medical laboratories are not accredited. Now, obviously, we are not medical laboratories and therefore don't exist necessarily under the same kind of systems, but the implementation of a quality management system in the work we are doing is essential. And therefore, we've been working with Nadine and Barlab, which you'll hear a talk from later on, to implement a series of quality management practices to ensure that we have a consistent global transfer for the products and the techniques that we have. So we have a range of kind of SOPs and check sheets at present for that. But assuming that that's all fine and it does exactly what it's supposed to do, you're going to need to keep it and store it. While you're doing that, you can use your selection of buffers, salt, CDTA, reducing agents to do a variety of different purposes. But most of the time, most of the commercial reagents, and indeed any of you that have ever ordered anything, you'll probably get a nice polystyrene box from NEB in dry ice, and then you put it straight in the freezer. And then that's not always possible at various points throughout the world. Therefore, room temperature or just refrigeration only would be preferred. And the most common method that is achieved is via lyophilization or freeze drying. Freeze drying is or can be a complicated process where you have to consider a number of different aspects as seen here. And even if you consider those aspects, you still need to make sure that your enzyme or your protein, be it an antigen, antibody or whatever, is still stable. And to ensure that, you need to consider another set of considerations. So it's not easy. But we've tried to consider this as part of the ecosystem and have made a lyophilization guide, in this case for PCR enzymes, because that's a large proportion of the things that people want to produce. And yeah, provided simple kind of suggestions on what you can do to try to simplify the process. But large proportion of laboratories or facilities do not have expensive lyophilizers or anything like it, and therefore you still need a way to try to find out how you can preserve the enzyme for room temperature storage or shipping. And I would love to be able to provide you with a simple, you just need to do this. But unfortunately, most of the time it has to be empirically tested because there's so many different variables. It's unbelievable. So in conclusion, we provide you with all the DNA parts and or ecosystem enable you to perform the experiment you need, which is then able to be transferred and distributed and transferred and distributed. Ultimately, we are providing a self-replicating and self-empowering collection of tools and education. Thank you very much. Jenny, over to you.
Thank you very much. Great. So we've got time for maybe 2 questions. Okay, we have a question chat about the quality of the enzyme function. John, do you want to kick that one up briefly?
Quality of enzymatic function. That's an interesting one, actually, because somebody mentioned that when they produced fusion enzyme, it didn't function quite as well as the commercial counterpart. and that is something that we have to be aware of. Presumably by having a range of quality standards, you can kind of say, does it function as a PCR within certain parameters? If it passes those parameters, then yes. And in kind of commercial laboratories, in general QC laboratories, you have a quality control standard that it must meet, and presuming that you have the necessary controls, that the assay is running fine, as long as it functions as you've tested it, hopefully that should work.
Thank you, John. And we do have some SOPs for quality assurance tests on polymerases that are coming out soon, worked on by John and Nadine, who'll be speaking to us later about quality control. So I would highlight there is a lightning talk coming up